Microscopes are used for magnification. They make it possible to see tiny microorganisms, such as this amoeba. There are many makes and models of compound microscopes. The microscopes used in your laboratory might be slightly different than the one shown here. This video will describe the parts of a typical compound microscope, describe the care and proper handling of the microscope, demonstrate proper focusing technique, define total magnification, field of view, parfocal, contrast, and working distance, and describe how to improve contrast of a specimen. Using the compound microscope, magnification is achieved through the combined magnification of the ocular lenses and the objective lens. The bottom portion of the microscope is called the base. The base supports and steadies the microscope and contains the substage light or lamp for illuminating the specimen. The arm of the microscope is the vertical portion that connects the base to the head of the microscope. The intensity of the substage light is controlled by the light control knob or dial. The control may be located on the base or the arm of the microscope and is similar to a light dimmer switch. The microscope should always be carried with two hands, one holding the arm and the other supporting the base. The platform that the specimen rests on is the stage. Most stages have an attached mechanical stage that holds the slide in position and allows the specimen to be centered over the light source. Beneath the stage is the condenser, a non-magnifying lens that concentrates light on the specimen. The condenser often has two ways to adjust the light delivery: a knob that adjusts the height of the condenser, and an iris diaphragm that has a lever or dial to open and close the diaphragm. The height of the condenser is usually best kept closest to the stage. The height of the stage can be adjusted using the coarse and fine focus knobs. At the top is the head of the microscope, which attaches the revolving nosepiece and the ocular lenses to the arm. The nosepiece has a revolving mechanism that carries 3 to 4 objective lenses, which can be rotated into the viewing position. Light passes through the specimen and up into the objective lens, which magnifies the image of the specimen. Typically, a microscope has 4 objectives, each identified by its magnifying power, which is labeled on the lens: 4x, 10x, 40x, and 100x. The names on the screen are typically assigned to these magnifications. Note that the 100x objective is marked as an oil immersion objective. This means that immersion oil must be added between the specimen and the objective lens when this objective is in use. From the objective lens, light travels up to the ocular lenses, which are located in the eyepieces. Specimens are viewed by looking into the eyepieces. Typically, the ocular lenses themselves magnify the image another 10x. The total magnification of the image viewed is the product of the objective lens magnification times the ocular lens magnification. For example, if the 40x objective is dialed into place, the total magnification would be 400x. Compound light microscopes achieve magnification through a set of lenses that refract or bend light rays. Refraction by the lenses also creates image inversion, where the image is inverted both top to bottom and left to right. Notice that the magnified E is the mirror image and upside down when compared to the original. Microscopes are expensive, so caring for them properly is just as important as using them properly. We'll demonstrate typical tasks and techniques used in microscopy. Remove the cover and plug in the power cord, ensuring that the power cord is not a tripping hazard. Turn on the power button. Using only lens paper, clean the ocular lenses and the objective lenses using a circular motion. Always begin focusing with the lowest power objective lens in position, transitioning to higher magnification objectives as needed. At the lowest magnification, you have the largest field of view, measured as a field diameter. Place a slide on the stage. If the 4X, or scanning objective, is not already in place, rotate it into position directly above the specimen. The objective should click into place. If the objective is not fully rotated into place, you'll see a dark crescent shape instead of a full circle of light. Use both eyes to look through the ocular lenses of the binocular microscope. Move the ocular lenses so that they're a comfortable distance apart. As you look through the oculars with both eyes, you should see only a single circle of light. Move the stage as close as possible using the coarse adjustment knob. Move the stage down using the fine focus knob until you see the specimen. Then continue using the fine focus knob to sharpen the image. Adjust the light intensity to a comfortable level if necessary. We use contrast to describe the difference in the intensity between the specimen and its background. The contrast of the focused image may be improved by adjusting the microscope's illumination. Adjust the lamp intensity by rotating the dimmer knob. See how too much light or too little light can result in poor contrast and affect your ability to see the specimen. To further improve contrast, adjust the diaphragm by rotating its cuff or by moving the lever from side to side. Move the mechanical stage adjustment knobs to locate objects that are not in your field of view. Move a given structure to the center of your field of view. Rotate the nosepiece to bring the 10x objective into place. This increases the magnification of the specimen and decreases the diameter of the field. Let's check your understanding. Calculate the total magnification, or TM, when using the 10x objective if the magnification of the ocular lenses is 10x. Is it 10x, 20x, 100x, or 200x? Recall that the total magnification is calculated by multiplying the magnification of one ocular lens by the magnification of the objective lens in use. The objectives on most microscopes are parfocal. This means that when the specimen is in focus with one objective, it will be in focus, or nearly so, when a new objective is put into place. So only a slight adjustment with the fine adjustment knob should be required. You should also adjust the contrast of the specimen as necessary. Rotate the 40x objective into place. The working distance decreases with the higher magnifications because the length of the lens increases. Using only the fine adjustment knob instead of the coarse adjustment knob helps prevent cracking a slide with the objective. Let's check your understanding. Contrast can be improved by closing one eye when looking through the ocular lenses, adjusting the iris diaphragm lever and the light control knob, or rotating the ocular lens. Contrast can be improved by adjusting the intensity of the light two different ways, by opening or closing the diaphragm, and by using the light control knob. When viewing very small specimens, such as bacteria, blood cells, or sperm, we use oil immersion. Your instructor will give you detailed instructions when oil immersion is required. Prepare your microscope for storage as directed by your instructor. Proper storage usually involves the 4x objective in position, the cord wrapped and secured, the power turned off, a clean stage in the lowest position and clean lenses, and the dust cover in place. Microscopes open up a whole new world to us that's not possible to see with the naked eye. Learning to use microscopes properly is critical in many careers. Consider how forensic analysts use microscopes to compare evidence from crime scenes, such as the two hair fibers shown here.